Hi guys! In this video, we're going to explore Trichoderma, what it is, why it's important, and most importantly, how it's prepared for use. So, to begin with, let's understand what Trichoderma actually is. It's a living fungus and a type of bio-fungicide, mainly used to treat seeds and soil. Its role is to help control various plant and diseases caused by harmful fungi.
Trichoderma is a beneficial fungus that naturally exists in soil and helps control seed and soil-borne diseases when used as a seed treatment. However, since it's usually present in low amounts, it can't perform effectively on its own. That's why we grow it in laboratories and then introduce it back into the soil to unlock its full potential in supporting plant and crop health. The best part? Trichoderma is a biofungicide, making it a key component of organic farming practices.
Alright, now let's get into the step-by-step procedure of preparing Trichoderma. Materials required. Essential materials. 1. 20 to 25 conical flasks, for culturing and holding the growing medium. 2. 60-gram jaggery, acts as a nutrient source for fungal growth. 3. Yeast, helps in fermentation and growth of Trichoderma. 4. Distilled water, ensures a clean, contaminant-free medium. 5. Boiling utensil, to prepare the jaggery yeast solution. 6. Talcum powder, serves as a carrier material for the Trichoderma formulation. 7. Carboxymethylcellulose, CMC, used as a binder for the final formulation. You will also need some supportive materials for cleanliness and safety. And the essential item required is the mother culture of Trichoderma, which can be obtained from the nearest laboratory or purchased online. To begin with, we'll need several conical flasks. These should be thoroughly cleaned, properly dried, and then sterilized in a hot air oven until the temperature reaches 100 degrees Celsius. Once the glassware has cooled down, it is ready to be used and is set aside for culturing and holding the growing medium. For this project, we're using 90 grams of jaggery, and here it's being measured on the weighing balance. Once the jaggery is weighed, it's added to the utensil. Next, 15 grams of yeast extract powder is also measured and added to the same utensil. Now, a sufficient amount of distilled water is added to the same utensil. The mixture is then brought to a quick boil to ensure the jaggery melts completely and blends well with the yeast. Meanwhile, the autoclave machine is being prepared by adding a sufficient normal water for the sterilization process.
Five minutes later.
After some time, the jaggery mixture has been properly boiled and cooled. It is now being poured into a jar to adjust the quantity and maintain the correct proportion. We are here adding more distilled water for increasing the quantity of the mixture. The growing media is now being dispensed equally into all our glassware, as shown in the video, before being passed on for sealing with a cotton plug and a plastic wrap. And all of the glassware with the media is neatly wrapped and packed is placed inside the autoclave for the sterilization process. Once the tightly sealed and wrapped media is placed in the autoclave, it is set for sterilization. Typically, one whistle is sufficient, and the process usually takes around 30 minutes. After patiently waiting for about 30 minutes, we finally hear the much-awaited whistle, signaling the completion of the sterilization process. However, we still need to wait an additional 15 to 20 minutes for the autoclave to cool down enough to safely remove the glassware, followed by another 10 minutes to let the media reach room temperature before proceeding to the next step. Laminar airflow (LAF) systems maintain a sterile environment by providing a steady, uniform flow of filtered air to minimize contamination. They are also equipped with UV light to eliminate microorganisms on surfaces. Further, before use, the LAF surface is thoroughly wiped with a cotton soaked in 70% ethanol along with some necessary equipment like the burner, cork borer, and wire loop. Then the UV light is switched on for 2 to 3 minutes, during when everyone must leave the room. Next, the media is placed inside the laminar airflow cabinet one by one. Now it's time to introduce the mother culture. For this project, a selected subculture is brought into the lab. Just like other lab equipment, it must be properly sanitized before use. In this setup, 2 professionals are involved, one assisting and the other handling the culturing process. Each flask's plastic wraps are carefully removed and loosen the cotton plug for further process. With proper sanitation measures in place, our professionals begin the process. The subculture is gently rolled near the flame to prevent the contamination. Using the cork borer, small circular sections are cut from the subculture. During the whole process, the LAF unit blower fan is kept on. Each piece of Trichoderma culture is then carefully picked up and transferred into the media-filled flasks one by one. Here, the same procedure is carried out, but this time using an inoculation loop instead of a cork borer. Once all the flasks have been inoculated with the Trichoderma culture, they need to be sealed again, just like before. Each flask is carefully wrapped with plastic wrap and tightly secured using few rubber bands. This helps maintain a sterile environment and prevents any external contamination during the incubation period. After sealing, the flasks are transferred to the incubation room and placed there for the growth process. If the weather is cold, a heater or hot air blower is used to maintain a warm environment, as Trichoderma thrives in higher temperatures. This helps accelerate its growth and ensures effective development during incubation. After 3 days of incubation, the culture has started turning whitish, indicating initial growth. However, it still needs more time to fully develop. After 5 days of incubation, the culture has begun to develop a green coloration, showing further progress. However, it still requires time to fully mature. 7 days later, the culture has fully matured, displayed a rich dark green color. The growth is clearly visible, indicating that it's now ready for the harvesting process. We've now reached the harvesting stage. The fully developed Trichoderma culture is brought into the harvesting room. Each bottle is opened one by one and the contents are strained using a strainer, as shown in the video. After straining all the cultures, the mixture is taken for a quick blend to ensure uniform consistency. And here are the results after blending. This freshly blended Trichoderma mixture is now ready for the next step. In the next step, it's time to use the talcum powder. A total of 12 kilograms is measured and brought to the mixing surface, as shown in the video. The powder is evenly spread out to prepare for the addition of the blended Trichoderma mixture. Once poured, the two are thoroughly mixed together. To enhance the concentration, the remaining strained liquid from earlier is also added during the mixing process. Once everything is thoroughly mixed, the mixture is spread out evenly and left to rest for 1 to 2 days. After 2 days, it's finally time for the most awaited step, the packaging stage. Before we begin, 60 grams of CMC is measured using a weighing balance and added to the powder mixture, as shown. The mixture is then thoroughly mixed one last time before moving on to packaging. We're using 1 kilogram portions, packed into Ziploc bag. Once sealed tightly, each bag is labeled properly and the packaging date is clearly mentioned. And with that, our Trichoderma Bio-Fungicide is finally prepared and ready for use. After days of careful work, patience, and dedication, it's incredibly rewarding to see the final product come together. This organic solution will now go on to support healthier, disease-resistant crops, making all the effort truly worth it. Thank you for joining us on this journey. If you found this video helpful or have any questions, please leave a comment below.